Good afternoon, everyone. I think it's about time to start. Okay. So welcome, everyone. Welcome to the laboratory workshop along with the 28th Bangkok International Symposium on HIV medicine. So we now enter 28 years already for this conference symposium. So we are pleased to bring you in the laboratory workshop. This year we will have the topic about the innovative molecular diagnostic for HIV and opportunistic diseases. So we like to begin with the how molecular epidemiology in HIV. So it's my great honor to invite Professor Anthony Kelleher, Director of Kirby Institute, University of New South Wales, Sydney, Australia. Tony is a clinician scientist. He graduated from medicine at UNSW in 1986. He trained in internal medicine and pathology at St. Vincent Hospital at Sydney at the height of HIV epidemic, qualifying as a clinical immunologist and immunopathologist in 1995. He has played a role in multiple phase 1 to phase 4 clinical trials and cohort studies, exploring the treatment and prevention of HIV infection, especially in the conduct of correlative laboratory-based study. He has established novel assays for measurement of CD4 T-cell responses, several of which are patent and have been developed commercially and pioneered the use. For example, the use of fine needle biopsy of lymph node to study both immune responses and HIV reservoir. He is also exploring novel gene therapy approaches, including an siRNA-based block and box strategy for the control of HIV reservoir. Besides that, he is my primary supervisor when I did PhD at UNSW. So please welcome Tony. He will give us the learning about the how molecular epidemiology in HIV. Tony, please.
Thanks, Sassy. It's a great pleasure to be here again and to present at this workshop. And I hope that it will be interactive. If as we're going through, there are things that aren't clear, or if there are questions, please just alert me and we can stop the presentation and we can address questions as we go, or I'm happy to take questions at the end. And so I wanted to talk about molecular epidemiology of HIV and really how we're hoping to use that in Australia to help us end HIV and understand how the disease is currently spreading. Standard epidemiology is really important in understanding where we're up to in the epidemic, but there are things that standard epidemiology can't answer, and I think there are certainly molecular epidemiology is a complementary approach to that, and I'll hope to demonstrate that to you today. But it's far from simple to set up. There are all sorts of issues about setting this up, both in terms of community concerns, but also in terms of data governance and data ownership. And so I'm going to go through a lot of those very practical issues and sort of convey some of the experiences we've had over the last 3 or 4 years as we've tried to approach a national approach in Australia to this issue. So in the talk today, I'm going to talk about what molecular epidemiology is and really couch that in the context of how it might be able to help our public health responses and really focus those public health responses where they're most likely to be valuable, but also then as an assessment tool for whether those public health responses have been effective or not. So there are definite things that molecular epidemiology can tell us, but there are definite limitations around molecular epidemiology, and I want to make sure you all understand what those are. As I said, we've been trying to set up this as a national approach in Australia, and we've been doing this in our local jurisdiction, which is New South Wales, which is a state, one of the states of Australia. It's the state with the highest burden of HIV, and it happens to be the state where the Kirby is located, and we have a very good relationship with the public health laboratories there and the Ministry of Health there. So we've been working closely with them and the community groups to sort of try and establish this as a pilot site that we can then extend to the rest of Australia. And I don't think it's not so much the specifics of the issues we've had, but the principles which that experience has unmasked that I think are important for this audience. And I wanted to sort of show you where we were up to so far and what the challenges are going forward. So, as we all know, epidemiology is the study of distribution and patterns of diseases, and in this case, diseases caused by viruses within a population. Who's being infected, where are they being infected, when are they being infected? And molecular epidemiology adds genomic sequences to that, and genomic sequences, as we all know, are made up of the RNA or the DNA of the particular virus that we're studying, and in this case, that virus is HIV. And when we combine genomic sequences from the virus with specific epidemiological data, that then becomes what we call molecular epidemiology. And the sequence alone of a virus and how they're related will give you some information. You can tell whether there's clusters or not of related viruses, but it doesn't tell you anything more than that. So if you can annotate those sequences with clinical data, when people have been infected, what their demographic characteristics are, what their risk factors are, then you can understand much more about the drivers of those clusters and perhaps the networks from which those clusters arise. And that then our hypothesis is that will then give you a richer data set by which you can develop public health interventions that are much more specific and likely to be more cost effective. Molecular epidemiology has been widely used, so there's nothing particularly novel about what we're doing here, and it's been used across various viruses such as HIV, hepatitis C, and I guess where it became very widely known in the community around COVID-19, but similarly it's used to track respiratory viral infections and other respiratory viral infections and foodborne viruses and that sort of thing. So molecular epidemiology describes networks or clusters, and this is an example of that, and it tells you and it does that by telling you how closely related sequences are. So each of these dots represents a different sequence, a viral sequence, and you can see that a lot of the sequences sit within this sphere here, but there are some that are more closely related than others, and some of those form relatively small clusters and some of those form much larger, tightly knit clusters. And so these are much more closely related than these, and these sequences here have no direct relationships. So it allows us, we can use the sequences and how closely they are related to develop a picture of whether those sequences cluster together or not. So clusters are what we sample, but we can only sample from people we know that are HIV positive. There are clearly people that are not diagnosed, and so while a cluster tells us about the relatedness of sequences within those that have been diagnosed, they represent potentially a larger group of individuals that form a network that are undiagnosed. And so this diagram here shows you that there are various forms of virus and the patients they come from within a network. So there are those that are diagnosed and for which we have a genome available. There are those that are diagnosed but we don't have a genome available. And then there are those that are undiagnosed and those that are at risk of HIV. So within that network. So the molecular epidemiology will give you an insight to what this network will, through the clustering, will give you an insight to what this network might look like, but it's not going to give you the full picture. But as the data becomes available, that the view of these clusters might change over time, and you can look at how the cluster is, or the network is growing over time, and what the drivers of that might be. And so studying the clusters give you the insights into what networks might be present, and because there are clusters of correlated infection, it tells you where the gaps are in your public health intervention. There are populations that you're not getting to, that you're not preventing transmission in. Similarly, if there's a lack of clusters, that potentially represents successful intervention. So someone's been infected but hasn't passed it on to another person, so there's no cluster there. That suggests to you that maybe public health interventions might be working. It might actually be a false positive for that, because you may not have a sequence from the people that have been infected in that network, but it may be a signal that your public health interventions are actually working, and I'll come back to that later. And so this information can be used to supplement standard epidemiology about what public health interventions are working, what aren't, and what public health interventions might be developed. So there are things that molecular epidemiology can't do. So molecular epidemiology tells you that two sequences are related. These two sequences, the blue and the yellow sequences, are related. They're closely related. The genetic distance between these two sequences is only small. But whether this person has infected this person or vice versa, you can't tell from molecular epidemiology. You can't tell whether there's been as the case here, that there's been a person that hasn't been sequenced, or a virus that hasn't been sequenced, that potentially infected both these people, or that there's been a person or a person in between these two people, or in fact several individuals between those two infections. So all it tells you is that the sequences are related. It doesn't tell you the direction of the transmission, or that the transmission has been directly from this person to this person. And that's really important to understand, and a really important message to get across to your colleagues in public health, but also the colleagues you're working with who are in the community. And we shouldn't be using it for that sort of purpose. And so as another way of ensuring that that's the case, the way we've decided to do this is we use genetic distance, which just tells us how many base pairs are different within the total sequence at each site, and that gives us an idea of the genetic distance, which is always non-directional, against phylogenetics. And with some very sophisticated phylogenetics, you can impute potential direction of transmission. This is, we've taken this approach of using molecular distance rather than true phylogeny to try and comply with UNAIDS' position on the use of phylogenetics. It's their position that phylogenetics can never be used to prove the transmission between two individuals. And we think by using this genetic distance, we reduce the likelihood of that happening further. And I think that's an important message that we've got across to the community groups with which we work. The other reason to use the molecular distance rather than true phylogenetics is that it's much more computationally effective, and it's as useful as phylogenetics for population-based interventions and assessing those. So just to define some terminology here, when we look at the relatedness of sequences, we can see sequences that are on their own and not related to any other sequence, and we call those singletons. We can see groups of sequences, or two sequences that are related to each other, and we call those pairs. And then anything with more than two sequences, we call clusters. And for our purposes, and I'll come back again later this when I'm actually showing you some real data, we've defined a large cluster as greater than 7, but you can use whatever number makes sense in your particular epidemic. But we've chosen, when I refer to clusters, that's more than 2 sequences that are clustered together, and large clusters greater than 7 sequences. So by doing molecular epidemiology, we can rapidly detect new variants, as we did in COVID, and we can also monitor the transmitted drug resistance and whether that's occurring by transmission of closely related drug-resistant sequences. As I said, we can use this to map networks of transmission against demographics, against risk behaviour, against where these people live or where they're diagnosed, and other demographic characteristics. Importantly, what it can do unequivocally is it can exclude that one person has transmitted a virus to another because the sequences are completely unrelated. And if you look at the definitive papers that show that U equals U works, because remember those studies were done often in pairs of individuals where there was one individual that was infected and one individual that wasn't. And you looked at the intervention to see whether the uninfected person became infected. And obviously that person could become infected by their partner in that relationship, or they could become infected from outside that relationship. And the molecular epidemiology showed that the transmission in those treatment as prevention trials occurred outside those relationships. And so it was very, very important in terms of showing that treatment as prevention works. And so while it will never tell you that one person has given you, definitively one person has given the virus to another, it can tell you that two people that might think they were infected by each other weren't, right? And as I said, the clusters we detect tell us about gaps in our public health response. So if we're going to get to virtual elimination, we need to understand at a much more granular level where transmissions are continuing to occur. And in some at-risk populations, we have a much poorer understanding of why transmission is occurring in those populations. And if there is ongoing transmission within particular populations, then you can, and there's clustering of those sequences, then you can look at public health interventions that target those particular at-risk populations, including developing culturally appropriate messaging about testing and treatment. You can, as I'll show you, there is in Australia, and particularly in Sydney, there is great unevenness in the effectiveness of public health interventions across at-risk populations. In Sydney, we've virtually eliminated transmission of HIV in inner Sydney, which is an extraordinary achievement given that that's where the epidemic in Australia started. But in the outskirts of Sydney and in regional Australia, we've made virtually no difference at all. And understanding what's driving the transmission in those populations is very important. And I hope to show you towards the end that molecular epidemiology will help us understand what interventions are needed on top of what we already know from the standard epidemiology. So at the moment, a lot of the new diagnoses in Australia are from recently arrived people, and what is unknown is whether they're forming networks where those recently arrived people are forming a cluster, or whether they're not transmitting the virus, and there's just a higher incidence of HIV in those recently arrived people who are arriving HIV infected. And again, that's a question that the standard epidemiology in New South Wales, although it's very sophisticated, can't answer, and we can answer that question, and I'll show you the answer to that question later in the presentation. It allows us to identify strain-specific and transmitted drug-resistant patterns, but the timeliness of this data is really important, and a lot of the regulatory and ethical and legal parameters in which we work really slow down the collection of this data, issues about data ownership, and we've really tried to address that and speed this up so that it's more real time, and we're getting the answers to this at the same time as we're getting the answers to the traditional, we're producing the traditional epidemiological reports. And one of the issues in Australia is that we have notification of HIV, but that gives you demographic data only. The sequence data is completely separate, and while it's standard of care that everybody who's newly diagnosed gets a sequence, we have to find ways of putting those two pieces of data together. And so a lot of our work has been looking at that. And we need to bring all these ideas together for a cohesive, more timely national data collection to improve our public health responses to go for elimination. As I've just said, there are complex regulatory and legal landscapes in Australia, but those, while they're different in detail, they exist everywhere. There's complexity around data governance and data ownership, and in our case, the separate holding of the genomics and the metadata causes issues with the data ownership. There are huge inefficiencies in current data practices. There are complexities around data linkage, where that data is stored, how it's protected, how the ownership of that is accepted. And then there are significant ethical considerations and community concerns around privacy, around data security, around transparency, and around, as is the problem in Australia and across this region, the criminality associated with transmission of this virus. And so linking sequences makes the community quite uncomfortable because they're worried about possible prosecution. So we formed this consortium called H2SEC, which was looking at bringing together both molecular epidemiology for HIV and hepatitis C. I'm really going to just speak about HIV in this presentation. And the aim of this consortium was to develop a national system for real-time molecular tracking of new HIV diagnoses and integrating that sequence data with risk factor, age, disease stage, location of infection, and those sorts of things to annotate the sequences. And the purpose is not just to do this to have a look at it, it's really to then produce reports that are accessible to public health units that they can understand, that gives them rapid data on where new and growing transmission clusters and networks exist, so that they can then, along with us, look at developing new public health interventions for what is, in our case, an increasingly sporadic infection. And so that, and then as we do that over time, that should tell us what's working, what's not working, where the gaps are, and we think it's an important component of achieving virtual elimination. So one of the challenges for us is, it's a great advantage, but it's also a challenge. The sequences are collected for patient management. So every new diagnosis is supposed to have a sequence done to tell us about the prevalence of drug resistance and to make sure the first-line therapies that we've started in these individuals are appropriate. But because that's patient-specific information, it can't routinely be used for public health purposes. And certainly it can't be used in an identified way for that purpose. And so we've had to address the legal and ethical implications of using that data that's there and available, and remember, is high-quality sequence data because it's been produced for personal diagnostic use, and so it's done by public health laboratories. It's very high-quality data, so that's a great advantage. But there are limitations within the law and the regulations about how that sequence can then be used for public health interventions. There is, although it's high quality sequence, the areas that are sequenced, the methodologies that are used vary from pathology service to pathology service, and we've needed to account for that. And we started this as a research project. We had to use the central linkage mechanism within New South Wales for data, and that was highly inefficient. Thank you. As I'll show you, and we've had to develop and get approved by the Ministry of Health new ways of doing this that are more efficient. So the H2SEC partnership is, that one? Okay. Okay, thank you. The H2SEC partnership is a large partnership, and it includes not just academics like myself and clinicians, but public health and reference laboratories, each of the state public health departments, the peak community groups within each state of Australia, and including the national ones, and as I said, a range of academics. We've really concentrated on making sure that the sequencing is high quality and that we've got QA and QC embedded not just into the sequencing but into the development of the reports. We've got specific working groups working on that, on data handling and analysis, on how the public health responses will be evolved from these reports. And then we have a pan-national strategic advisory group to help us with the national rollout of this eventually. And so the partners are shown here, and you can see that it's a big group of people working together, so huge collaboration. So as I said, we originally started this as a research project in 2015, and we were linking this from the notification data. We don't get a name, we get a 2x2 name code, we get date of birth and postcode, and we extract those from the sequences as well. And then we link on those parameters, the name code, the date of birth, and the postcode. What we report out has no identifiers. We don't report out specific age. Again, what we're trying to do is make sure people can't be re-identified. So we report out age in groups of 5-year blocks. We don't report out the suburb in which they live. We report out a statistical area, which is based on postcode. We report out risk group, disease stage, gender, the region of acquisition, their country of birth, and at the moment we don't include Indigenous status. And so what we're trying to do is develop an accurate near real-time linkage and notification data and sequence data, and then to produce highly reproducible, quality-controlled quarterly reports, which is what the standard epidemiological reporting in New South Wales achieves. And we want to incorporate this into the standard epidemiological reporting processes in New South Wales. And the analytics platform we're building in compliance with various ISO regulations. The object is to describe populations at risk and networks, populations within networks, not individuals, and to make sure that the data presentation can't re-identify those individuals. So what have we managed to do so far? So we've reviewed, the thing about Australia, it's a federation, so each state has a different notification process and reports different notification data. We've managed to standardise that. We've got an in-depth understanding of the legal frameworks in which we work across each jurisdiction. We've conducted extensive stakeholder consultation to understand the complex ethical considerations and issues around data ownership. And we've established a virtual network of public health units, diagnostic, molecular virology, laboratories, researchers, and community. We've optimised full genome sequencing methods, particularly for hepatitis C, which are not standard. And we've verified those, and we've instituted quality assurance programs within the participating labs. We've developed and tested an efficient data linkage mechanism that efficiently irreversibly de-identifies people. And we've developed a decentralised data sharing model where only the reports, not the raw data, are stored centrally. And we've completed a proof of principle clustering analysis, which I'll show you at the end of this presentation. So we've developed this model where we can pull the sequences out of each of the labs that does the sequencing and simultaneously pull the notification data from the public health units and link those in the cloud, and then generate the linked data set, and then generate a report, and only the report is stored centrally. So none of the raw data, none of the raw sequence data actually ever leaves the laboratories, and none of the raw notification data leaves the public health unit and is held anywhere centrally. So we call this a decentralized data management model. And that's required quite a bit of computer know-how. And so what the Kirby, who leads this effort, holds is the report, and that's all we hold. So even if you get the report, you can't work backwards to get to the raw data. The raw data is held with the original data owners. And again, this gets around some of the data ownership issues. We've shown that so the way we do the linkage, as I said, we link on these 2x2 identifiers. It's not showing up. These 2x2 identifiers on date of birth, and then this goes through a hashing program that generates this long sequence of numbers, of random numbers and letters, that is now the identifier. And so this is called a hash, and this is a two-step process where we have the hash and then we have a salt, which then re-randomizes the sequence of identifiers that goes to each patient, to each linked data set. And so each individual has an identifier generated by two what we think are irreversible processes of hashing and salting. And so this is, again, trying to make sure that people can't be re-identified by authorities such as the police. With this linkage process, it's very efficient. We get about 87% efficiency in terms of linkage. When we did this through the old methodology, which I won't go through here, we were getting about 70% accuracy at linkage. So significantly better than the standard approach recommended by the authorities. So what's the current situation in Australia? So as I said earlier, we're doing reasonably well, but we could do better. So there's a downward trend in new diagnoses made in Australia over the last decade. So in 2015, there was 1,030 cases each year. In 2024, there were 757. That's nationally. In New South Wales, which is where we did our pilot study, those numbers have reduced from 350 to 238. The main risk factor is men who have sex with men, and this represents about 60 to 70% of the new diagnoses. Both treatment and PrEP are freely available, and that's not just to Australian citizens, but to recent people who have recently arrived in the country. We showed that it was economically efficient to treat those people, even though they don't have Australian citizenship or what we call a Medicare card, and usually don't have access to the universal health care. Those people have access to both subsidised antiretroviral treatment and also PrEP. There's very high treatment uptake among new diagnoses and very high uptake of PrEP within most MSM. And we've had a 60% reduction in the rates of infection in Australian-born men who have sex with men. And we're on track, as I said, for virtual elimination of HIV within the inner suburbs of Sydney by 2030. However, as I said, there's been virtually no reduction in the rates of infection in men who have sex with men who are born overseas. No reduction in the rates of infection in those who have recently arrived in Australia, and no reduction in heterosexual spread. And a lot of our diagnoses are still late, 38%, and this is especially in the regions of Australia outside the main centres and on the outskirts of places like Sydney. And this is especially in those that have been born overseas. So there are clear gaps in the public health messaging and access to PrEP and other prevention strategies. So as I said, we did this pilot study in New South Wales where we took all the sequences from 2017 to 2023 and linked those by our decentralized linkage mechanism. And you can see that over those years we had rates between 70 and 80% in terms of linkage. And we link just on the first sequence that's available for each individual, because obviously people, if they're failing therapy, may have subsequent sequences done. So we just take the first sequence that's available in each individual and link that to the notification data, which gives us the demographics, the risk factors, and the stage of disease at which they were diagnosed, and also their postcode. So what have we seen? So there's been a steady decrease in the proportion of sequences that are subtype B, and the relative proportion of sequences that are non-subtype B have increased. The second most common subtype is AE, and then we can also track over time the sequences, the other subtypes, and you can see there was this spike in 2003 of subtype F, which actually happened in a cluster. So they were not separate importations of subtype F. We can look at drug resistance, obviously, and so in these sequences that are associated with new diagnoses, there were no mutations to integrase inhibitors. There were no new diagnoses with resistance against tenofovir, which is what we use in PrEP along with FTC or 3TC. And there were 3 diagnoses in which we saw the M184V, which confers high-level resistance to FTC and 3TC, but there were no sequences that had both tenofovir and FTC or 3TC resistance. We can see the imprintation of non-NUCs resistance mutations, and there is ongoing spread of high-level resistance against NRTIs over time, but that's not used as a first-line therapy in Australia. So we looked at 3,600 sequences between 2004 and 2023. And just taking the large clusters first, so these are all the large clusters we saw, that is more than 7, so there were 45 of those. And these show you when those infections were diagnosed over years. We calculated the growth rate and the cluster size as the total number of diagnoses within clusters, and the largest cluster contained 28 sequences. So just drilling down on that a little bit more. So firstly, you can see that these clusters, these ones here already existed, right? In 2000 and before 2014, but have continued to grow over literally over a decade. And so clusters can span decades. There can be great periods in which clusters don't grow. So they can appear dormant for years. You can see that in this cluster here, there's a gap of about 8 years between these 2 sequences here, but that's not on its own. There are other areas where, there can be gaps of 2 to 3 years in terms of when new sequences are added to clusters. But many clusters appear to have terminated, and they're shown here. all these sequences have terminated, and this is in the era as we introduced PrEP very effectively into the MSM population. And so you can impute that perhaps PrEP on top of TSA, in terms of on top of treatment as prevention, has contributed to the termination of these clusters. However, as you can see from here, there are still active clusters. There are 13 clusters. Most of these started despite PrEP being available. These clusters, the clusters with higher growth rates appeared in the last 6 years of the observation period, and that's despite the fact that there were fewer diagnoses during that time because of the COVID-19 lockdowns. And so there's clearly active local transmission and growing clusters despite the public health interventions at PrEP and treatment as prevention, easily accessible treatment during this period. So there are clearly gaps in our public health intervention. So what do those active clusters look like? So we can look at those by disease stage of when they were diagnosed, and you can see that the majority of these were diagnosed early, but there was a significant number of late diagnoses within these clusters. The risk factor is mostly men who have sex with men, and most of the large growing clusters are not from people that have come from overseas. They're people who were born in Australia. So the epidemic does not appear to be driven by the importation and then spread of infection from elsewhere. It is being driven by people born in Australia. While the area where there's a large contribution in some of these clusters to people who are living in regional Sydney, the clusters are spread across all regions of New South Wales, both the inner city and the outer parts of Sydney, and also regional New South Wales. So there is obvious mobility within the population, which again may not be surprising, but the growth of the infections in regional and the outskirts of Sydney is not being driven by transmission that is strictly within those suburbs or those regions, but is more diffusely spread, probably because of the mobility of people. So we can drill down on these in more detail. You can see that this cluster here is a mixture of people who were born in Australia or born overseas. You can see that the cluster has grown over time, but there are the people who were diagnosed come from all parts of Sydney, all the different LHDs, the local health districts within Sydney, and so there's not, as I said, geographical clustering, and in fact here there is this cluster represents is driven by both Australian-born and overseas-born men. So we can look at this in detail, and just to summarise, we can look at where we think the infection occurred, where these people were born, whether they've arrived recently or not recently, whether they were diagnosed early or late, and where they lived. And the synthesis of this information was that the singletons, which I haven't talked about, the majority of the singletons are in diagnoses with people who are born overseas or with overseas acquisition. So the importation of the sequences into Australia are not driving new clusters. In fact, those people remain as individuals who are not infecting other people. So that importation of sequences is not driving the ongoing transmission of the viruses in Australia. The large clusters, in fact, have a higher proportion of diagnoses among people born in Australia, and none of the actively growing recent clusters are driven by people who are recently arrived. So what we've seen in the singletons and what we've seen in the large clusters complement each other and cross-validate each other. The stage of infection is evenly distributed across the diagnoses in the large clusters, and the large clusters have a relative overrepresentation of people from regional New South Wales and outer Sydney, but many of the clusters start from infections within inner Sydney and then spread from beyond there. So we think we've made a range of outputs that are important in terms of public health. We've identified that there's a large number of active clusters that are there despite the public health interventions. The growth rate of those clusters is not decreasing despite the public health interventions. There are still numerous late diagnoses within these clusters, and the clusters are widely geographically spread. So targeting public health messages to particular geography is not likely to be efficient. One of the questions that the health department wanted to know was whether people arriving recently were spreading the disease among themselves, and I think we've definitively shown that that's not the case. So the community had a range of concerns about this approach around the misuse of data, particularly around criminalisation. And I think I've shown you that we've tried to mitigate this in a range of ways by using molecular distance, so that rather than phylogeny, we've gone to great lengths to de-identify the data and make sure that those data can't be re-identified. And we've come up with a way of linking the data and having the data not move out of the places where they're owned, and only the report being centralised. And therefore the report can't be hacked and disentangled because there's no data held with the report. It's held at centralised facilities. We've introduced safeguards to protect the data from law enforcement, from criminal legal courts, from immigration enforcement, and even if that data were obtained, re-identification would be impossible. And we think we've minimised costs by making sure that we're using data that's already collected and available, making the maximum use of both the notification data and the sequences that are held by the laboratory. So at the moment we're in the final processes of developing a report that's accessible to public health professionals. This is what our dashboard will look like. We will map various parameters, including where the infections are occurring within the state within the last year, the growth rate, and this will all be interactive. And we hope to be able to develop these reports and make them available on a 3-monthly basis. And I'll leave it there. Thank you.
So thank you so much for Tony for the very informative information. I think if we may have the question will be yeah, okay.
Thank you very much. Very informative discussion, and I feel it was very complex topic, but you broke it down wonderfully. But I'm still stuck. You started with cluster, you defined singleton, then moved on cluster and networks. You didn't talk about networks. Could you share a little bit what is the statistical relationship, how you develop network on top of cluster?
So I guess network is a term, and we haven't addressed that. So really, I mean, our analysis ends with the clusters, and then we can impute networks from those. We haven't developed the statistical approach to, I guess that's at the moment beyond what we're trying to do. But, you know, you can use, so at the moment we have estimates from molecular epidemiology about, from standard epidemiology about how many undiagnosed people there are in Australia, right? Or in each jurisdiction. So we take a statistical approach to that. We can use these data to look at that in a different way, to see if we get the same sort of numbers in terms of undiagnosed, and that would then, that statistical approach would then inform us more about the networks. But frankly, there's so much regulatory and particularly regulatory and data ownership issues to develop that we haven't spent a lot of time doing the sophisticated analyses we'd like to do. So that's, hopefully, as we've got this now embedded and we've got the reporting sort of sorted out, we can do some more of the sophisticated statistics on the data sets to sort of estimate the size of how large those networks might be, those clusters, and whether, in fact, the undiagnosed are actually more likely to be associated with clusters, or whether they're likely to be undiagnosed singletons, right?
Thank you, Jenny. That was an excellent story. My name is Raf Hamers from Oxford University, based in Indonesia. I was curious if you can say a bit more about the community opinion. You said there were concerns about confidentiality. After doing all of this, starting the process and having first data, have you gone back to community organizations and showed to them what you are doing with them? And did that change perspectives? Did it provide reassurance?
So the approach we've taken is, they were part of the consortium from the beginning. I spoke briefly about the working groups we had. So we have a working group that's on sequencing, a working group that's on the analysis, and then a working group on the public health interventions. And we had community, we had community representatives on those working groups, so they could see what was happening as we developed each of those things. So we've tried to keep, it wasn't a model where we get them on board and then we come back to them at some time later. They're actually part of the process. And I think that's been important in terms of bringing them along with us, because there are, not just in Australia, but across the communities of people living with HIV, there is great concern about how molecular epidemiology is going to be used. Even over time, the level of acceptance of this waxes and wanes a bit within the community. At the moment, the community is very keen to contribute to the virtual elimination of HIV, and so they're actually on board with this. But they do put significant parameters on what we do and limitations on what they would do. And in fact, they will, at the moment, their position is that they would actually like named linkage, but they'll only allow that to happen if the federal government and the state governments decriminalise transmission. So they're sort of almost using this as a bargaining chip. So I guess the short answer is they're embedded in the process, and the community is not a uniform group, as I'm sure you're well aware. And so, we have to go back over the same ground repeatedly. So, just before Christmas, myself and Leo, who's been doing a lot of the linkage, etc., did another presentation to the peak national group of people living with HIV and AIDS. We did a 10-minute presentation and then did a half-hour discussion where we just took questions to try and make sure. So it's not just the people sitting on the working groups who are, speaking for community. We speak to wider groups of the community, and I think that's been a really important part of making sure we keep the community on board with what we're doing. Because if we don't have them on board, this is going to be, it's really difficult as it is, it will be impossible. So, I guess, just on the data ownership, I've shown you the data on 2023. I can't show you the data beyond that because the Public Health Department in New South Wales hasn't approved me to discuss the data beyond that, right? So there are all sorts of sensitivities both within the community and within the public health departments about what these data tell us, right? So there's a lot of expectations and concerns to manage, which is actually, the linkage is quite sophisticated now, but not, and that virtual, doing that on the, sort of virtually is really, really clever, I think. It wasn't my idea, but it's been really clever and owns a whole lot of things. But, the most difficult thing about this is managing the expectations and the concerns of not just the community, but the public health departments, the data ownership of the sequences by the laboratories. It's managing all those issues that actually slows this down, right? Rather than the actual technicalities of actually doing this stuff, right? These reports, once you've got the stuff, you can generate now in sort of minutes, right? Takes a lot longer to analyse them, but it's actually managing all those competing interests and concerns that's really the difficult thing in doing this, rather than the science and the analytics.
Well, thank you so much, Tony.